The massively transformative purpose of AcuraStem is to bring curative therapies to all of the patients that have neurodegenerative diseases and who are going to have neurodegenerative diseases. AcuraStem is focusing at the outset on ALS, not only because it's an incurable and devastating, fast-acting disease, but it's primed for a breakthrough from the scientific standpoint.
ALS in particularly is devastating because it affects people ages between 35 to 50 when they're in the prime of their life and work life. Traditionally, scientists has been studying ALS that focus on a small population of ALS patients that has a particular genetic form and heavily rely on animal models to study this disease. And that's explained the reason why there is no treatment for this disease. And the current available drugs in the market, Reluzol and Radicava, can only extend patient lives for a few months. That's why we're focusing on ALS.
To understand what we do, I often draw an analogy to the oncology field, where they biopsy patient tumors and study them, individuals' tumors in a dish. So we do the same thing in neurodegeneration, but the challenge is you cannot biopsy brain cells or nerves from the central nervous system. So step one in our platform, our technology, is that we can take people's blood cells or skin cells and turn them into living neurons in a dish. So these are people's own cells in a dish that we can study. So that's part one. Part two is to understand what's the disease characteristics in the dish. And so what we've done is take a lot of, generate neurons from a lot of healthy patients and a lot of ALS patients, and then quantify the differences to come up with a disease signature.
Layered on top of that is the ability to look at those cells and see what's happening to them and see which ones are neurons and which ones are surviving over a period of time and to see how they're changing and see what synapses are forming and see what branching is occurring. And to be able to analyze that and quantitatively display that, that's cutting edge. I think that's cutting edge. It's like the Google Maps of motor neuron biology.
And we can use that to find new potential therapeutics that seem to reverse these processes in the nerve biopsies. But in principle, this is applicable to all of the neurodegenerative diseases.
We're also collaborating with biotech companies and pharma companies who are studying and developing ALS drugs to see if their lead compounds can rescue any of these phenotypes in the patient motor neurons. So this approach is truly revolutionary, and we hope to apply to this not only to ALS, but in the future to other neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease.
What we're doing is really revolutionary, and that there's real hope that we might generate really actionable information for their clinician that could improve their quality of life. So we're not talking about helping future patients. We might be able to help today's patients.